Okay, so I decided to record a new updated PowerPoint on cell organelles for my AP students simply because I was noticing some confusion and um, like misconceptions or lack of knowledge, and I think that falls on me. So therefore, I've added a little bit more detail to my cell organelles PowerPoint. So here we go. When we talk about cells, we can divide cells on Earth into two separate categories. We have prokaryotic cells and we have eukaryotic cells. Now we'll talk in detail about the two different types of cells and what they have in common and how they are different. But generally speaking, prokaryotic cells are going to be bacteria and archaeans. Now, when we look at these pictures, we can see they look very similar and they have quite a bit of things in common. So when we say prokaryote, we could be referencing either bacteria or archaeans, and there's only slight differences between them. Now, when we talk about eukaryotic cells, these are going to be our larger cells that contain organelles, like membrane-bound organelles, as well as a nucleus. Like their DNA is contained within a nucleus. And so we will also talk about eukaryotic cells. But first, let's go ahead and look at some common characteristics that both bacteria and archaea share. So these are characteristics of prokaryotes. So they are single-celled organisms. Now, if you come across a word problem that mentions a unicellular organism or a single-celled organism, it doesn't necessarily mean that it's a prokaryote. There are eukaryotic cells that can be a single cell, like paramecium, euglena, amoebas. They're all single-celled eukaryotic cells. So you got to look for other clues in the problem or in the wording to see, do they have organelles? Do they have a nucleus? Etc. So single-celled organisms, all prokaryotes though are single-celled. You are not going to find a multicellular prokaryote. So all prokaryotes are a single cell and they have one large circular chromosome floating in the cytoplasm. Their DNA is not located within a nucleus. They lack or don't have membrane-bound organelles. When we say membrane bound, what we mean is an organelle. Let's take like a lysosome, for example. The surrounding part, like the outside of that lysosome, is composed of a lipid bilayer, the same material that our cell membranes are made out of. So prokaryotes lack membrane bound organelles, and they have ribosomes. All cells on Earth have ribosomes. Ribosomes are organelles. However, they're not membrane bound. They're actually made out of rRNA and protein. And then they have plasmids. Plasmids are these extra little pieces of DNA that we'll talk quite a bit about in genetic engineering, as well as a bit in our discussion on mitochondria and chloroplasts. Anyway, and they also have a cell wall that surrounds their cell membrane. So when we talk about like slight differences between bacteria and archaeans, we can see that the bacteria cell wall that is found outside of the cell membrane is made of peptoglycan. And so peptoglycan, I think I said that wrong, is made of both protein and carbohydrates that you can kind of figure out from the name, versus the archaean cell wall is made of polysaccharides. So one minor difference, but when you look at the DNA between the two types of prokaryotes, we see that the archaeans' genes are more similar to eukaryotic genes. Now, I also want to point out, though, on Earth, if you look at all the prokaryotes on Earth, the majority of them are going to be bacteria. Archaeans are also known as extremophiles. You will find archaeans in extreme locations on Earth. For example, the like hydrothermal vents or the hot springs in Yellowstone National Park, where I believe is where they were first discovered. Or you might find them in like really salty environments, like salt flats or something. Okay, so let's go ahead though and talk about some characteristics of prokaryotes. So when we look at my image that I have here, this is a general structure, but if you look along the bottom, you can see that there are different shapes of prokaryotes. Now, all prokaryotes will have one large circular chromosome, and then this plasmid is an extra like hoop or circle of DNA that is not their main chromosome. It's a little tiny like extra DNA like double helix that is in a circle. Okay. Then we have, they all have a cytoplasm. When we see the word cytosol versus cytoplasm, Cytoplasm is the space within the cell that kind of surrounds the DNA, plasmids, ribosomes. But when you say cytosol is the fluid in the cytoplasm. So I kind of use the words interchangeably. But anyway, so you might see cytosol, you might see cytoplasm. Now, here we have ribosomes within prokaryotes. Now, this here, cilia and flagella, are not unique to prokaryotes. There are eukaryotic cells that have cilia and have flagella. So they are involved in locomotion or movement. So the flagella would help if this bacteria kind of like swims or something, this flagella would help it move. We see this in sperm, for example, have flagella as well. Now, prokaryotes, another difference between prokaryotes and eukaryotes is that prokaryotes, when they multiply, so if one bacteria or one prokaryote becomes two, the process is called binary fission. It's a type of asexual reproduction where they basically make a clone of themselves. So this is different than a eukaryotic cell that would divide by mitosis. Okay, so let's go ahead though and zoom in on this prokaryote. I want to talk real quickly about cell membrane versus cell wall. Now, when we look at this image, we can see the cell membrane in gray, and it's surrounded by a cell wall. Now, a cell wall and a cell membrane, those two words are not interchangeable. They mean different things. So if you are talking about a cell membrane, make sure you do not accidentally write, like in an essay or something, a cell wall. They have two different purposes, and they are two different structures. The cell membranes surround all cells, and then all prokaryotes have a cell wall on the outside of that cell membrane. Okay, so now though, let's go ahead and compare eukaryotes to prokaryotes, and let's see what they have in common. So things they have in common, all cells are surrounded by a cell membrane, or a lipid bilayer, or a plasma membrane. They all mean the same thing. They all contain ribosomes. So eukaryotes as well as prokaryotes have ribosomes to carry out protein synthesis, and they all have cytoplasm. Now, I do want to point out that plant and fungi have cell walls. Plant cells and fungal cells have cell walls. So their cell membranes are going to be surrounded by cell walls. However, animal cells do not have cell walls. It's nice that our cells are flexible. Think about our red blood cells as they travel through our circulatory system. It's nice that our cells are flexible. So I want to be clear. If a cell has a cell wall, that alone does not tell you if it is a prokaryote or a eukaryote. You'd have to look for other clues to deduce, is it a prokaryotic cell or is it a eukaryotic cell? Okay, so let's go ahead though and talk about eukaryotes now. So in eukaryotes, they have linear chromosomes. So the prokaryotes had a circular chromosome. We can think of it like a necklace, like a hoop, versus linear chromosomes. I like to picture them as shoelaces, right? Linear chromosomes are located within a nucleus. That nucleus is membrane bound. And then they have organelles that are surrounded in a lipid bilayer. They can be multicellular or unicellular that we talked about. Now, plant cells, compared to animal cells, have a cell wall made of cellulose. They have chloroplasts that are not found in animal cells, and they have a large central vacuole. Now, I do want to point out something students mix up is that mitochondria are actually found in both plant and animal cells, as well as fungi and protists. And so I kind of want to say all eukaryotes have mitochondria. It's not unique to animal cells. I think the misconception shows up where we think of photosynthesis as an animal process. Oh my gosh, as a process in plants, and we think of cellular respiration and mitochondria as like an animal thing. But I want to point out that plants carry out photosynthesis and cellular respiration in their mitochondria. Okay, so let's go ahead though and now go through the different organelles. So here we have a nucleus within our cells, and eukaryotic cells. This is where we find our DNA and our linear chromosomes. Now the nucleus is surrounded by a nuclear envelope. That nuclear envelope is a lipid bilayer. There are things called pores located within that nuclear envelope. I like this picture particularly. If you've taken the DNA out, right, you can see the little pores within that nuclear envelope. Now you also see this like folded area around or right next to the nucleus. So this area here is the endoplasmic reticulum, our next organelle to discuss. Now, when we look at my little cartoon drawing of this cell, this eukaryotic animal cell, the ER is located here. So you have the nucleus in purple, and then you have the smooth ER in yellow, and then the rough ER in blue. So the smooth ER's job is to synthesize lipids. So we can think of triglycerides or cholesterol or phospholipids. And it also has enzymes that are involved in detoxifying the body. So whether it's drugs or harmful chemicals or alcohol, the smooth ER will help to detoxify and break down those toxins. So, for example, a person who tends to drink a lot of alcohol, over time, the cells in their liver will have larger and larger smooth ERs to handle that toxin coming into the body. Then we have the rough ER. Now, the rough ER, when it was first looked at underneath a microscope, it looked a bit rough. And so that's where it gets its name, rough ER. Now, why it looks rough is because it has ribosomes attached to the surface. So if you look at my picture in blue here, those little brown dots are ribosomes, and we call those bound ribosomes. They are bound to the rough ER, and they make proteins. Now, when we talk about proteins that are made on the rough ER, they will eventually exit the cell, and so I have a few slides coming up about that. Now, when we talk about ribosomes, though, ribosomes make proteins. So protein synthesis happens at a ribosome. Now, ribosomes have two parts. They have a large ribosomal subunit and a small ribosomal subunit. So when you look at this and it looks like a hamburger bun, that top part is the large subunit, and then the bottom part is the small subunit. They're found in all cells, both prokaryotes and eukaryotes. Now, the size is different. In prokaryotic cells, the ribosomes the subunits are a bit smaller. Function, still the same, but the size is a little bit smaller. Now, I also want to point out that in eukaryotes, the ribosomes can be found in two places. They can be free floating within the cytoplasm. You can see in both my pictures little brown dots spread throughout the cytoplasm or spread throughout the cytosol, and they are bound to the rough ER. In both my pictures, you can see the little brown dots attached to the rough ER. Those are ribosomes embedded within that lipid layer, that lipid bilayer of the rough ER. Now, the next organelle that has something to do with proteins is the Golgi apparatus or the Golgi body. Now, the function, this right here is the Golgi, and I also want to point out it's also found in plants. Now, I do want you to notice, though, that the Golgi and the rough and smooth ER both have these folds. Now, even though they're a lipid bilayer like folded, we'll notice that the Golgi is separate from the nucleus, the nuclear area, right? It's not attached to the nucleus like we see with the rough and smooth ER. So it is separate out in the cytoplasm. Now, the function of the Golgi is basically to package, modify, and sort proteins that were made by ribosomes on the rough ER. The Golgi also plays a role in producing lysosomes, another organelle we'll talk about. But now let's go ahead and look at the different organelles that are involved in the production and export of proteins. So this is called the Endomembrane System, and this will be helpful if you are taking a biology class. When you learn about protein synthesis, this is the path of how it occurs. So in our nucleus, we have our DNA. Now, our DNA is what contains the directions for how to make proteins. That's what our genes are. But the DNA is stuck in the nucleus, and the ribosomes are outside of the nucleus. So we need to get the directions on how to make a protein to a ribosome. So we make RNA in a process called transcription. So RNA is going to be a copy of DNA that is made in the nucleus. This process is transcription. Now, the RNA will leave through a nuclear pore. It's going to find a ribosome, a bound ribosome on the rough ER. Now, that bound ribosome is going to then read the messenger RNA, and it's going to synthesize or build a protein or a polypeptide. And so I have it here in purple for you. That's a cute little polypeptide made by the ribosome. Now, it's going to be carried in a transport vesicle. So that little blue circle you see there, You can think of it like a taxi for the cell. It's going to carry the polypeptide from the refiar to the Golgi. Now, the function of the Golgi apparatus is to package, modify, sort the proteins that were produced on the refiar. Now, I want you to think about this. If you are in AP Bio and you've already learned about macromolecules, we learned that a protein is a polypeptide that then undergoes a secondary structure and a tertiary structure and possibly a quaternary structure. And it has a final shape, because shape determines function. This finalization of the shape is going to occur within the Golgi. Then the protein, if it's meant to be exported from the cell, it'll get like a little, you can think of it like a little address label attached to it. So it is like sent out from the cell. So here we need somehow to get the polypeptide or the protein from the Golgi to the cell membrane. So this is where another transport vesicle is going to come and basically pick up the protein, and then it will carry it to the cell membrane, where now that protein has two possible fates, sorry. That protein can either become a membrane protein, like a protein channel or a receptor protein or an enzyme within the membrane, or it can be sent out of the cell by exocytosis. Now, when this transport vesicle is interacting with the ER, the refiar, and the Golgi, and the cell membrane, it's actually fusing. The lipid bilayer is fusing with the lipid bilayers of the organelles and the membrane. And so very similar to endo or exocytosis. Okay, so let's go ahead then and talk now about, we have just a few more organelles. So here we have the lysosome is next. So the lysosome is probably my favorite organelle, and the lysosome is involved in digestion and waste removal. If you have damaged mitochondria within your cell, they're going to be broken down and recycled by the lysosome. If there is a cell that engulfs, maybe like bacteria or pathogens, like a white blood cell, that bacteria or pathogen will be digested or hydrolyzed and broken down by the lysosome. Or if the cell is like an amoeba, like a single-celled eukaryotic cell, and it engulfs and takes in food, that food will be digested by the lysosome. So let's go ahead and see how that works. So here I have, let's pretend it's a white blood cell. And so we have our lysosome, and on the inside, within that lysosome, there are hydrolytic enzymes. If you've already learned about macromolecules, we know that hydrolytic is referencing hydrolysis, which means to add water, to break, or to separate. So here we have our hydrolytic enzymes that when this white blood cell engulfs the bacteria, we can see that cell membrane forming around and bringing that bacteria in. This process is actually called endocytosis. And now that black circle around that bubble is actually a lipid bilayer. And the lysosome is made of a lipid bilayer. So when the lysosome comes in contact with that engulfed bacteria, the two lipid bilayers will fuse. And then those hydrolytic enzymes are now in the same vacuole as that bacteria, and it can be degraded. So that is how the lysosome works. All right. So now let's go ahead and talk about two energy-producing organelles. We have the mitochondria. Now, the mitochondria generates large amounts of ATP. Hopefully, in your biology class, you'll have a whole unit on cellular or aerobic respiration. They're also called the powerhouse of the cell. They are a cool organelle that has a double membrane, and they carry out aerobic respiration in different regions of the organelle. Now, if you're in AP Bio, you'll also learn that the mitochondria evolved by a process called endosymbiosis, which is a whole separate video. But let's go ahead and look at the structure of the mitochondria. So the mitochondria has two membranes. It has an outer mitochondrial membrane as well as an inner mitochondrial membrane. Now, the matrix is the area within that inner mitochondrial membrane. This is where, when you get to aerobic respiration, we'll learn that's where the Krebs cycle happens. Then the main parts of mitochondria that generates a lot of ATP is going to be in that inner mitochondrial membrane. There's these things called electron transport chains that are responsible for the mass production of ATP. And so those are located within that inner mitochondrial membrane, but you'll get a whole fun unit on that topic in the future. Now, the second organelle that has two membranes and is involved in energy is the chloroplast. Now, the chloroplast is only found in plant cells, and so, or algae, I guess too, right? And so here, I want to point out, you can see there's mitochondria also in this plant cell. But here, the chloroplast is where photosynthesis happens, and it's going to absorb sunlight, and it will take carbon dioxide and water from the environment, and it's going to take that C, H, and O and rearrange it into macromolecules. In this case, we have glucose as our example. And so the chloroplast carries out photosynthesis. But when we zoom in and look at the structure of the chloroplast, again, you'll study this in more detail when you get to your photosynthesis unit. However, within the chloroplast, there's these membrane-bound thylakoids. Now, this is where the light reaction happens. So in these thylakoids, there are pigments. So when we say the sun shines on the plant and the plant absorbs that solar energy, it's happening here in the thylakoids. Now, a stack of thylakoids is called a granum. And then the space outside the thylakoids is called the stroma. The stroma is where the Calvin cycle occurs, and that's where glucose and our carbohydrates are produced out in the stroma. And then finally, our last organelle we're going to talk about is our vacuole, or a vacuole. And in plants, it can take up to 50% of the cytoplasm. It stores a lot of water. It can store pigments, or even be like sort of poisonous, like as toxins accumulate in the cell, they can like dump it into the vacuole so it's not harming the organelles. Now, vacuoles are also really important in turgor pressure to like press on the cell wall and help plants stand upright. All right. So sorry, it's kind of a long video, but that is my new updated summary on organelles, and hopefully it goes into enough detail for you. Great job.